The Wisconsin Fast Plant was created by the University of Wisconsin-Madison professor Emeritus Paul H. Williams as a research tool that could ultimately improve disease resistance of cruciferous plants such as cabbage or radish. In order to create a plant with these capabilities Professor Williams was looking for, he had to breed Brassica rapa and 6 related species from the family Cruciferae for shorter life cycles. After 20 years of experimentation, the Wisconsin Fast Plant was finally a reality, turning what was a 6-month growing process into one that only took 5 weeks to complete. With an average of 15 centimeters in growth height and a life cycle of only around 40 days, the Brassica rapa was finally complete in its development and is now used for many types of experimentation. By definition, evolution is a process by which different kinds of living organisms are thought to have developed or diverged from earlier forms during the history of Earth. Now with evolution comes artificial and natural selection. Natural selection is the natural reproductive process where the strong survive without human interference. Artificial selection is the process in which humans pick a desired trait to breed a living organism in order to produce more offspring organisms that will genetically obtain the same traits as the parent organism. This selection process may have multiple steps that are taken in order to see the desired trait in future generations. The only difference between artificial selection and natural selection is the human decision-making factor that takes place in artificial selection. Artificial selection has been around for thousands of years, with the early stages coming in the field of agriculture, yet Charles Darwin noticed the selection by humans and coined the term artificial selection. One of the main purposes of trichomes on plants is the protection they provide from animals and insects. These unicellular or multicellular mechanisms can also differ in the amount of protection in which they provide. Some trichomes can actually accumulate harmful chemicals that can be used on organisms looking to disrupt the plant. Trichomes also cover the body of the plant. This is because the division of the plant's epidermal cells, which causes the spread of trichomes on both the leaves and stems of plants. These trichomes also help to maintain normal levels of radiation from the sun and help the health of the plant by reducing the loss of water. From reducing evaporation, keeping frost away from leaf cells, and protecting against harmful organisms, trichomes are very important to the success of many plants.
Our purpose is to prove that we can acquire the desired traits through artificial selection, in this case, a higher trichome count per plant in Generation 2 than in Generation 1. Our experiment was performed to answer the question, can fast plants be artificially selected for a greater amount of trichomes? In other words, does human nurture affect the growth of trichomes in fast plants, or is it simply the amount of trichomes depending on nature due to the genes of the plant? Our hypothesis is that if fast plants that are not in the top 10% of the highest number of trichomes are taken out, then the subsequent generation of fast plants will retain an increase in trichome count per plant from generation to generation as a result of artificial selection. To set up this experiment, first fill 4 planting pots halfway with soil. Water the soil until it is completely wet and let it drain through the holes at the bottom of the pot. Evenly spread out 6 fast plant seeds per pot and then proceed to lightly push down the seeds into the soil, about 2 millimeters deep. After, place each pot into a greenhouse where artificial sunlight can shine on the plants 24/7. Within the next couple of days, roots and sprouts should be visible for almost every seed, and the plants will have started their photosynthetic processes. With steady watering on all weekdays, the plants were measured for their heights weekly. By the end of the second week of Generation 1, our tallest plant was already 8 centimeters, and they continued to grow in height as their 4-week life cycle progressed. Before the fast plants reached the near end of their lifespan, we pollinated the plants. To do this, we used Q-tips to pick up pollen from one flower and rub it onto another plant so seeds are produced. After pollinating, we stopped watering the plant once we saw seed pods and allowed the plants to dehydrate so the seeds were in condition to be planted. These seeds created the plants for Generation 2.
This is the data from the first generation of fast plants. There was a total number of 173 plants in this generation. The highlighted numbers were the winners, while all the others were losers. The losers were either removed from the pots, or if there was a pot with only losers, they were banished to the loser section. The next set of data was for Generation 2. In this generation, there were 73 fast plants. There were no winners or losers in this generation, only because we did not need to pollinate to make a new generation. The first generation statistics, which were taken on day 14, were 7.84 trichomes as the mean, 4 trichomes as the median, a range of 59 trichomes, and a standard deviation of 11.03 trichomes. The Generation 2 statistics taken on the 14th day as well was a mean of 7.6 trichomes, the median was 3 trichomes, a range of 62 trichomes, and a standard deviation of 10.94 trichomes. When both generations were combined, there was a total number of 246 fast plants, with both generations combined. The mean was 7.79 trichomes, a median of 4 trichomes, a range of 62 trichomes, and a standard deviation of 10.91 trichomes. Because there were 100 fast plants in the first generation, obviously it would seem that the first generation had more trichomes in all categories. But in order to determine which generation created more trichomes, we must find the percentage of each category. In the generation, when this is done, one can see that the second generation created a higher percentage of higher amounts of trichomes, but as a whole, the difference of trichomes was minimal.
We planted the fast plants on August 20th for the first generation. For the second generation, we planted the fast plants on October 22nd. In the first generation, we did 6 brassica seeds per pot, but on one of the pots we did 7. And then in the second generation, we did 5 brassica seeds per pot, but on one of them we did 6.
While taking care of our fast plants, we noticed several things. The most notably was that the generation 1 grew at a much faster rate, as well as the first generation had more plants per pot than the second generation. Even though we only planted 1 more seed per pot for the first generation, only 1 or 2 plants actually sprouted in the second generation per pot. This trend of generation 1 growing at a faster rate than the second generation continued into week 2 and on. As you can see from the comparison of day 27, fast plants, generation 1 had much taller and more plants than the second generation. In conclusion of the observations, although the first generation grew at a higher rate and had more successful amounts of plant sprouting, it did not affect the amount of trichomes for generation 2.
Based on class data obtained over the course of 4 months, we can conclude that our hypothesis was not correct. The observed data we obtained suggests that even though a few plants had more trichomes in the 41 to 70 range in the second generation, the fast plants did not produce enough trichomes overall to prove that the plants were hairier in the second generation. In fact, only 3 plants produce a visibly higher amount of trichomes in the second generation than the first generation. Also, the mean number of trichomes per plant of the first and second generations were only different by 0.24 trichomes. Such data is conclusive in saying that there is not statistical evidence to suggest that more trichomes were made in the second generation than in the first generation. These observations can be reasoned due to the fact that certain genes were turned off in the second generation. Even though the second generation plants contained the genes to produce more trichomes per plant, during the winter when the second generation was planted, the plants were affected by the decrease in temperature. This decrease in temperature caused the trichome gene to essentially turn off. This could be attributed to fast plants not needing to defend themselves as much during the winter, since their predators are often in a state of hibernation.
One of the errors that was encountered within our experiment was uncontrollable temperatures. These temperatures ultimately could have affected total growth of the fast plants, resulting in a restricted amount of trichomes that could grow on the plant. Ideal temperature for fast plants to grow is between 72 degrees Fahrenheit and 82 degrees Fahrenheit, and the plants did encounter temperatures outside of this range. We could have corrected this error by growing the fast plants in a more temperature-secure room with less fluctuation and not a greenhouse with thin walls and inconsistencies in lighting and heat. The greenhouse the plants were in also may have had differentiating humidities that also would have affected the plant's growing ability. The humidity affects water vapor on leaves of plants, so low humidity will affect growth differently than a high humidity would. Adding a humidifier to the room to control the humidity would allow for a common humidity during the entire growth of the plants. The last error that the fast plants may have encountered was the inability to naturally photosynthesize due to the growth in an enclosed atmosphere. The fast plants were unable to go through natural photosynthesis in an open atmosphere, and as they grew, they needed to photosynthesize more. Ventilation into the greenhouse may have allowed for a more natural photosynthetic process for the plants as they continued to grow, because they would have had a more regulated atmosphere.
As a group, there were many successes that we saw in the second generation. Although we were unable to create hairier fast plants in the second generation, we were able to increase the amount of fast plants with a trichome count between 41 and 70. Some failures that continued into the second generation were the inconsistency of plants growing after the seeds had been planted. This could be due to the way in which we planted the seeds into the original soil. There was also the failure of every plant that did not produce trichomes. In generation 2, it was expected that plants would all produce trichomes, yet statistics were not much greater than that of the first generation for plants that didn't produce trichomes.